to speak about parasitic plants, especially to groups of enthusiasts like yourselves. I've been working on parasitic plants pretty much my whole career. And tonight, my talk is going to focus on the parasites that you find in New York. They're fascinating biology and just some things that demonstrate their natural beauty too. Sometimes people have a disconnect between the idea of a parasite being beautiful, but I think if you look at that Agellinus on the title slide here, who could argue that that isn't a beautiful wildflower? And yes, indeed, these are parasitic plants, as is the daughter, Cuscuta, maybe a little uglier than the Agellinus, but still fascinating. So let's go ahead without any further ado. And we're going to, right off the bat, let's get some terminology out of the way, just because I'm going to be using these terms. By the way, how do I get rid of the thing that's blocking? I see everybody's picture across the top, and it's blocking out part of my slide. Is there a way I can put it on the right or something? Let's see. Hide video panel, is that? Oh, that got rid of some of it. Okay. So what we're looking at here is the blueberry order, the order Ericales. And you guys all know blueberries, the genus Vaccinium. This one over here, that's a beautiful one from the Philippines, by the way. And what we're interested to know, though, is are there fungi associated with their roots? And in many cases, Vacciniums do have mycorrhizal fungi, as you know, many of our forest trees do too. Another member of the family, little Chimaphila maculata, it's also autotrophic in that it has green photosynthetic leaves, but it clearly is a myco-heterotroph. It has fungi associated with its roots that help it feed. If we take that to the next step in the family Ericaceae, the blueberry family, I'm sure many of you probably have seen this one, the Monotropa. It's a beautiful ghostly white, completely non-photosynthetic myco-heterotrophic plant. And it's kind of nice that this picture shows it next to a log with some shelf fungi because its roots are intimately associated with mycorrhizal fungi. So this is truly a heterotrophic plant, a different feeder. It's obtaining nutrients, not just water and minerals, but also other things from the fungal associate. And finally, this weird thing over here on the left, Mitrostemon. It's a true hostorial parasite, and we'll see what that hostorium is in just a second here. This picture was taken in Japan. It occurs through the Philippines and Malaysia, and then strangely enough, another species in Chiapas, Mexico. How about that for a distribution for Mitrostemon? It's a, again, completely non-photosynthetic holoparasitic plant. So this is what my talk's going to focus on today, is the holoparasite, or the parasites, not so much mycoheterotrophs. So what is a hostorium? That's what we're looking at here, is a whole bunch of different ones. This is one from the sandalwood order in the genus Ximenia. You can see the hostorium is light in color. Here's its connection to this sort of flanged portion. And that's the host root, the brown part. And then all these are all various different ways that the parasites attach to their host. This Balanophora forms a tuber, a potato-like structure, that here's the poor host root coming in, and here's the poor host root coming out, not much left because all those nutrients are going into the parasite. Canophilus, we're going to talk about, is a native in New York. So we're going to see that one, as well as Arcithobium. So we'll talk about their hostoria when we get there. So again, the hostorium, a modified root, forms the morphological and physiological link between the parasite and its host. By the way, if you guys have any questions or whatever, please feel free to just chime in, because I kind of get rolling here. All right, let's see. I'm going to show video panel over there on the left. Okay, that's better. Ooh. Okay, so a little bit more terminology. The two flavors of parasitic plants that we have are hemiparasites and holoparasite. I've already used that term, holo, which means total. Hemiparasite is a bit of a strange word that's sometimes abused. It doesn't mean that half of the plants are parasitic and the other half aren't, or half of the plant is a parasite and the other half isn't. It simply means that of the two sources of basically nutrients in a plant, the xylem and the phloem, right? These guys only attach to the xylem. Hemiparasites are xylem water feeders. Holoparasites not only get the xylem, but they also get the phloem contents, the sugars, amino acids, and all of those things. And that's why they can abandon photosynthesis. But in the hemis, you can see, hemiparasites, you have a variety of different modes, trophic modes, from facultative to obligate. This means that these facultative ones can actually grow without a host. You can stick them in a pot and grow them, and they'll use a host if you give it to them, but they don't really need one. Whereas obligate, they have to have a host to complete their life cycle. And that's a beautiful mistletoe shown there. In fact, all of these are mistletoes except for the Lauraceae down at the bottom here, Cassytha. This is one of our strange members of the laurel family in the tropics that looks like dodder, like Cuscuta. And then over here, that's the actual dodder or Cuscuta that we'll talk about in just a bit here. And all of these groups are holoparasites. So that's evolved in about 9 times in flowering plants, where we've gone all the way to being holoparasitic. And some of the strangest plants on Earth. And I've worked on some of these, Hypneraceae, Rafflesia, so on. So these are they're topics for other talks. We're talking about New York parasites. I wish we had Rafflesia and Hydra, but we don't. Another way to look at the hostorial parasites is to plot them on a phylogenetic tree. So this is sort of our concept. This is my new mouse. This keeps, I move it and it does that. This is the arrangement of orders, if you will, within the flowering plants, from Amborella all down at the base, all the way up to the Lamiales that we're going to talk about today. And in red are all the cases where we have hostorial parasites. So immediately you can see parasitism has evolved multiple times independently, in fact, 12 times. That was the topic of my paper, Parasitic Angiosperms, How Often and How Many. It looks like that we have actually 12 independent evolutions of hostorial parasites. And that's what we see over there with the letters that are keyed to the tree. Okay, so we're going to be talking about just a few of these today. So in New York, this is what we have. We actually have 5 families, 21 genera, and 40 species of hostorial parasites in the state of New York. The vast majority of them are in Orobanchaceae. As you can see, a lot of generic diversity. And then in my favorite group, the sandalwood order, we actually have 3 families for these guys over here with 5 genera. All right, so we're going to have a look at each of these separately. So we'll start off with maybe the most familiar one to you guys that maybe you've encountered dodder before, the genus Cuscuta. It's hard to really go anywhere in the world because there's some very weedy ones. And we're kind of starting off way up at the top of the tree here in the Solanales, the same order as potatoes and tomatoes and petunias. All right. So here's a beautiful native dodder I took in the Great Smoky Mountains National Park, Cuscuta rostrata, and you can see it twining around like spaghetti around its host and then forming these pretty little white flowers. When you dissect the flowers, you can see the petals and the stamens, but there's also this structure. They're called scales. And it turns out that the morphology of those scales is important in the taxonomy of dodder. My friend up in Canada, Mihai Kostia, works on dodder, and he has a whole web page. He takes beautiful close-up photos of dodder showing the morphology of those scales. So a lot of the taxonomy depends on floral dissection. In terms of parasitism, this shows the hostorium of dodder entering into the host. So there's the host epidermis, here's the vascular tissue, and then the cortex in the middle. And you can see it just goes right in there, and it affects well, plumbing to plumbing connection, if you will, right? It literally connects up to the individual cells and gets nutrients that way. Dodder is sort of tottering between holo and hemiparasite. A lot of them are true holoparasites, where they have pretty much abandoned photosynthesis. This is an African one, by the way, Africana, very robust dodder from there. So let's have a look in New York, though. This is what we have. Start off with Cuscuta campestris. Oh, this is becoming very annoying. It's going forward on me. I'm going to not touch the mouse, maybe. I will leave that alone, and I'm going to use the keypad. Yeah. All right. So one of the most common daughters is Cuscuta gronovii, the one you see on the right over there. In Southern Illinois here, it's blanketing the banks of every major creek, river, and so on, and major lake. And you can see in New York, it's very common. So this is where my mentor, Lytton Musselman in Virginia, he says the parasitic spaghetti. It does look like someone dumped a bowl of spaghetti. Daughter, it's a strange plant. It germinates, and then once it establishes connection with the host, it twines around and grabs a hold and forms a hostorium. It gets disconnected at the ground. It has no more connection to the soil at all. So you can actually take a little daughter and throw it on your unfavorite neighbor's plants, and it will grow. No, I'm not recommending that. A lot of these daughters are named for the host that people originally thought they were specific to. So Cuscuta coralli, the hazel daughter, it does occur on Corylus, but it's not specific to that one. So that's one of our native ones. It's, as you see from the map, scattered in New York. Cuscuta australis, very uncommon in the state, southern daughter. A lot of these are more common outside of New York. It's not that it's rare and endangered everywhere in its range. And then these are non-native ones down at the bottom, by the way. They're introduced. So epilinum, upon linum, linum is flax. So that's that one. And epithymum on thymus, right? So this is a European one I've actually seen in Spain, but it's been introduced to New York. We don't have that one in Illinois here. So I'm going to have to go out and see your Cuscuta epithymum. The epilinum one is an interesting, I show a picture of its seeds there. There's a mutation that forms a double seed. You can see some of them are doubled. Why would that be important? Because then it's pretty much the same size as a flax seed. And the machines that separate the flax seed don't then separate out the daughter. So it's often transmitted with its host when people plant flax. So it's gotten all over the world this way, and people have actually published some interesting papers about the evolution of that mutation with people growing flax and the daughter adapting to that. So that's Cuscuta epilinum. Some state rare ones now. So these are ones that are truly, they're native and they're truly rare. Cuscuta campestris is not rare elsewhere, but it is apparently in New York. This one is found worldwide. It's one of the most common daughters. Some people make it synonymous with Pentagona. If you visit the USDA plants webpage, they have that all wrong. They have to read Mihai's paper where Pentagona is a tetraploid versus campestris is a diploid. They're separated on the phylogenetic trees. They have different chemistry. So older works may have lumped them together, but those two look similar, but they are distinct species. Cephalanthi, buttonbush daughter. This is one we have in Southern Illinois. We have lots of buttonbush. I noticed they plant a lot of buttonbush on the Ithaca campus. Maybe we need to get that one going on it. Compacta, you can see where it gets that name. The compact daughter, sometimes rope daughter. You see it forms these dense masses like twining around as rope around its host. Looks like it's only down there in the southern part on Long Island and so on. Obtusiflora, glandular daughter, Pentagona, and Polygonorum. Yes, it probably parasitized the polygonum originally, but most of these things have broader host ranges than that. A guy named Joe Neal, Pennsylvania, grew a lot of these in the greenhouse, so you can see his name. I'm crediting the photos for him, and he cultivated them on coleus and all kinds of different exotic hosts. All right, so that's daughter. That's our first common parasite, and sometimes not so common for the state of New York. All right, so let's move on to the next order. This is the Lamiales. It's a very big group. You can see up at the top there, it's near the Solanales. And you probably learned some of these as Scrophulariaceae. But these days, with molecular work and other things, Scrophulariaceae has been disintegrated. In fact, that's a good talk for this group, right? What happened to Scrophulariaceae? Because it's now in multiple families, and one of them is Orobanchaceae. It used to be Orobanchaceae meant the holoparasites only. But now Orobanchaceae includes all the hemiparasites as well as the holoparasites. And you can see that on the phylogenetic tree on the right. Those are all the genera in Orobanchaceae. It's a big group. And the ones in green are hemiparasites. The ones in yellow are holo. And immediately you can see, holoparasites are not all on the same branches, right? The same clades. That means that they've evolved multiple times independently of losing photosynthesis. And we're going to actually have a look at a couple of them down at the very bottom there, Epiphagus and Conophilus in clade A, way down at the bottom. This group includes some of the worst parasites in the world in terms of human agriculture. So we'll have a quick look at some of the nasties, the good, the bad, and the ugly, if you will. But what's interesting is the one shown at the bottom there, Lindenbergia. I'm going to risk using my mouse here. This one down here. This occurs in the Philippines, actually. It's a non-parasitic member of Orobanchaceae. And where does it occur in the phylogeny? Way down here, along with Rehmannia and how do you say that? Trienophora, the Chinese things. So those are non-parasites. So this is really why people now are using those as a model to study how did parasitism evolve? Can we actually find some genes, in these more derived ones that are not present in the ones from which they gave rise to them? So that's why people are interested in Lindenbergia. But I told you I would take a little diversion from the good, the bad, and the ugly. These are the bad. This is Striga asiatica. I did my master's degree on it. Why? Because it was introduced in the 1950s into North Carolina to about 20 counties. What does it parasitize? Corn, maize. And here's Striga growing nicely on the roots of corn. My work did some anatomy. I actually sectioned flowers and I showed that they are autogamous. They pollinate themselves before the corolla even opens up. And that's what we're showing here. Corolla lobes closed, pollen right on the stigma, and you get the capsules. Each capsule makes about 5,000 tiny, tiny little seeds, almost the size of orchid seeds. Here it is compared to a corn, Caryopsis. So they're really good weeds, right? They don't need a pollinator, and they make tons of seeds that can last. They tested them about 20 years in the soil. They can even take freezing cold temperatures. They mimicked Ames, Iowa. So the real question when I was working on this in the 1980s, why hasn't Striga spread, right? Why didn't we get Striga into the Midwest? We're just lucky, I guess. But in sub-Saharan Africa, this is the #1 cause of crop loss over insects and everything else. Striga, which means witchweed, it bewitches the host because you don't get any crop after that. And Striga is a very interesting plant in terms of studying how it finds its host. Turns out that it was originally from cotton. They isolated this molecule called strigol. Then they found it in the root exudates of other plants. And when the little seed of Striga is pre-treated and exposed to strigol, it comes to life. It starts to germinate. And it forms this radicle or little baby root. And then it senses another host exudate that they've later called xenognosins, and it starts to differentiate these root hairs. And those are the hairs that actually stick the little baby seedling onto the host root. And you can see it in this nice scanning electron micrograph here. There's the seed coat still present. Here's the little root, and there's the poor host root. And it's sticking it on there. Later, it forms a primary hostorium, and the little baby Striga is off to the races. This is all happening underground before you even see that your host plant, your crop, has been parasitized. When it gets older, this is what the hostorium looks like, penetrating into the host root, attaching to the xylem. So Striga was federally controlled. Nobody was allowed to move it and work on it, willy-nilly in your lab back when I was a graduate student. So my mentor, Lytton Musselman in Virginia, got a guy named Jim Riopel at University of Virginia, Charlottesville, to pick another plant. He picked a native one called Agellinus to study because it exhibits the same phenomenon of recognizing these exudates. And these are just some slides showing you the time frame. 18 hours after induction, meaning after you've given it the host exudate, 3 days, 6 days, until finally here it is attaching to the host root with those hairs. This hair has been pulled off, and look at it, brought some of the cell wall of the host with it. That's how good the glue is. So Jim Riopel and his group prompted others, John Yoder and David Lynn, and eventually they worked out the whole recognition process. I won't go through, there's a lot of chemistry here, but basically these plants are reacting to these exudates through a redox signaling pathway. That's actually what causes the cells to start differentiating the hostorium on the root. And then it attaches, and again, we have a parasitic establishment here. So it was all because of really working on a native plant, Agellinus, and that's going to bring us then to Agellinus in New York, because some of these are actually some of our rarest wildflowers. The very first one, Agellinus decemloba, sometimes called acuta, sandplain Agellinus. You can see it occurs just down there on Long Island. Only, I think, total of 15 total populations. This was one of the very first plants listed on the federal endangered species list. It grows in these sort of maritime areas that are, of course, being developed and also being infringed upon by Phragmites australis. So they're monitoring now a lot of these populations to make sure that they maintain them. So that's one of our rarest plants right there, Agellinus decemloba. Two other ones that like sort of wet situations is Agellinus marittima, the seaside Agellinus. I can see it's a little up closer to Lake Erie there. Maybe we could, I could go up and see. I've never seen this one, but I would like to go see it. Head south or north from Ithaca and you can get to that one. It says salt marsh false foxglove, but I don't think it's a salt marsh up there on the lake, would it be? Must take fresh water as well. Paupericola, the small-flowered Agellinus or small-flowered false foxglove. These things have a member of the acanthaceae and come up with another name, and Agellinus is what we have today. But the common names keep, like, I don't know, some of these have 3 and 4 common names. So those are some of our rarest right there. But we also have some pretty common ones. And Agellinus purpurea, the one that I showed as being used as a model organism, this is probably the most common Agellinus in Eastern North America. It's right here on our campus at SIU, actually. So you can see it's a couple places in New York there. Cetacea, the thread leaf false foxglove or needle-leafed Agellinus, it's scattered in a few counties in the state. And then finally, I'm sure one of the most common grows from New Mexico all the way to Canada and east. This is Agellinus tenuifolia, slender Agellinus, or sometimes narrow leaf false foxglove. And looking at the map there, you can see it occurs in half at least of the New York counties. This one took a little interest in Kurt Neubig, a professor shown there in his orange shirt and shorts a couple summers ago. We were advising Keegan Schultz. He was an undergrad and we wanted a project. So we thought we would use some a little bit of field work and a little bit of molecular biology to try to get an idea of the host range of this Agellinus. So we went to our nearby little Lake Murfreesboro, picked 5 sites where the plant occurred, and we went out and dug up plants. That's what we're doing right here, is excavating and bringing them back to the lab, washing them off really well, put them under a stereo microscope, and then look for the hostoria. And that's what this picture shows here. The brown is the host root and the white are the hostoria. But we weren't interested in the Agellinus, we're interested in what was it parasitizing. So we took a little clippet, put it in some silica gel, dried it, and then that can be used to extract DNA. We amplified a gene called ITS, one of the nucleoribosomal spacers, sequenced it, got the sequences back, and then did what we call blast. You throw those sequences against the huge database, and it tells you what your plant is. And lo and behold, we've identified the host, at least to genus. We also, by the way, made herbarium vouchers of everything that's in the vicinity at each of these sites. So here are the results. It occurs on a lot of different things. You see Plantago, Lesbideza, Antonaria, Fraxinus, but boom, Quercus. Lots and lots. It even grows on some monocots, but it seems to prefer Quercus. Now, the real question is it just because quercus roots were the most common in the rhizosphere or not? Well, not always. Sometimes the quercus roots were about equal to other ones, but this probably will take a more controlled study to actually document the total density of the different roots to really get to the idea that this thing has a preference for oak. But I think this is good first evidence that it does. And it also shows how good this technique is that could be used with parasitic plants all over the world where what's going on underground? We never really know, right? It's such a big mystery. We all talk about hosts, we look at what's growing around and we take some guesses, but we really don't know in many cases what these things grow on. So this method can help solve that problem. Now, another segregate of that old genus Gerardia is this one, Aurularia. These are some of, I think, our most beautiful wildflowers, these big bumblebee pollinated yellow flowers. Have you guys seen these in the field, right? Aurularia. This is a common one, Aurularia flava, the smooth false foxglove. This one, pediculaire, it has the sort of glandular leaves. That's very different than all the rest of them. They're kind of dissected and glandular. My mentor, Lytton Musselman, always asked, you know, which Aurularia is it in this pedicularia? It likes power line cuts. So we always find this in a power line cut. And then Aurularia virginica, first named by Pennell. Pennell monographed the Scrophulariaceae back in, I think, 1930s was his monograph. And he straightened out the taxonomy. These things were named all sorts of different things from Linnaeus on. So again, Aurularia, beautiful, beautiful wildflower. Other Orobanchaceae, the genus Buchnera. Scattered distribution from Texas all the way to the east and north into Canada. But as you can see, not that common in New York. And in fact, from what I've read, it's on the decline in most states, which is really too bad. This is called blue hearts. And you can see that it's being visited by a buckeye butterfly over here. And buckeye lays its eggs specifically on Orobanchaceae. There's some buckeye eggs on the calyx of that Buchnera. So this is one of our wildflowers that I would like to see sort of restored, brought back to some of our locations. Similarly, Indian paintbrush, Castelligia coccinea. It gets its name from the fact that the bracts are sort of red, like they were dipped in paint. Those are bracts, by the way. The flowers themselves are kind of drab, as you see here, kind of greenish. It's the showy part is the bract below the flower. And these are hummingbird pollinated. And of course, if you've traveled in the Western US, you see lots of Castelligia. There's dozens and dozens of species. It's very interesting. We really only have this one. There's actually a different color form of it, a yellow form, but not many species of Castelligia in the East. And it's, again, scattered in New York. And many of these, again, are declining populations. Cow wheat or Melampyrum, that's another one you'll see, but it's very common in New York. I need to get out and see this one in the field. I've seen it in Rocky Mountain National Park. But that's look at the strange shape for the leaf with that base with these pointed lobes. So Melampyrum lineare. And then finally, on the right down here, Schwabia americana shaft seed. This is probably one of our rarest North American plants. Occurs very, very rarely. Only one county I think it's reported in. I think it may have been extirpated from that in New York. Do any of you guys know whether Schwabia is still present in the state? I don't know. This is one I would like to, it's not too far away, go out and see if it's still there, see if we can relocate the population.
I think it's not, but it was recently rediscovered in Massachusetts after an absence of 100 years or something.
Wow. You wonder if it was in the seed bank or whether somebody brought it in, because it's being, the USDA seed bank does have Schwabia. Now, you can get some if you have a legitimate project. But of course there you'd have to worry about the genetics of where exactly it came from, because it does occur throughout down into the southeastern United States too. Yeah, Schwabia americana. Pedicularis, or the lousewort, wood betony. This is another one that goes by lots of common names. Very common in the eastern United States and New York, not in southern Illinois. Isn't that strange? We don't have Canophilus. This one is a parasite on oak roots. It really prefers oak woodlands. You can see the beautiful yellow galliate flowers here that are bumblebee pollinated. Here it is opened up section so you can see inside. The plant in fruit right here and the little capsules. These have little like diving board valve on the capsule. And this is a raindrop dispersed seed. The raindrops hit and bang, it springs up. It doesn't make that noise. I did that for special effects. But that's a case of a raindrop dispersed plant. And then you have one other species, the swamp lousewort. Pedicularis lanceolata. This one occurs in northern Illinois as well, but I've never seen it. So maybe I'll get a chance to. It looks like it's west and east, but not right where we are, huh? How many of you have seen that one? Anybody seen the swamp lousewort? Oh, you've seen all of these. Oh my goodness, we got to go out with you. Okay, more Orobanchaceae, but essentially everything you see here is introduced. I didn't realize New York had so many introduced Orobanchaceae. This is going to be fun to go out and find these. Euphrasia is a huge genus, over 300 species worldwide, occurs in Africa, New Zealand, Philippines, and New York. But this is one of them, Euphrasia stricta, the upright eyebright. Okay, cute little flower. The Orthocarpus, rosy owl's clover, I think they haven't really rediscovered it after its original introduction. So that may not still be extant in the state. I put it here anyway. This came off your website for the New York flora. So anyway, it's one to have a look and see if that one still occurs. To the right, Odontides, the red false Bartzia, it's occurring apparently as a weed all the way up into Canada. In fact, Canada considers that a noxious weed. So I haven't seen that one either, Odontides, introduced from Eurasia again. All of these are mostly Eurasian weeds. Rhinanthus, major and minor, are all introduced. These are very important plants in many situations in Europe. A lot of work has been done on the ecology of Rhinanthus in grasslands in the UK. Turns out that parasites are a good thing to have in your ecosystem. The studies show that when the parasite is present, it decreases the fitness of the major dominant members. Like in our case, prairies would be big blue stem and sorghastrum, the Indian grass. And if you have parasites there, like Castelligia or something, it lowers their density and it allows other plants then to colonize. So your alpha diversity goes up when the parasites are present. And that's been shown in England as well. So Rhinanthus, a good thing. Parasites are not always a bad thing to have in your ecosystem. It allows other things to have opportunity to grow there. What's weird is this Rhinanthus minor subspecies Greenlandicus. It's listed as native. So one subspecies, Rhinanthus minor, is an introduced thing, but Rhinanthus minor Greenlandicus is native. I have to check that out, huh?
Farther north, it's found.
Yeah, you see that county in New York, it's way up there, almost what, Vermont, right? Probably up into Canada, I think, right? So it just gets into New York right there. I don't know what the differences are taxonomically. There's probably some subtle, morphometric things on the flower dimensions or whatever to tell that one apart from the other subspecies. An interesting study for a student to see, what's going on. Okay, now we'll move from the hemiparasites to the holoparasites. Just two of them in New York, Canophilus, the squaw root, cancer root, oak drops. I hadn't seen oak drops before. That's on your New York website. And finally, Epiphagus, beech drops. So that tells you your host, oaks and beech, for the two different parasites here. I'm sure all of you have seen Canophilus, or it's hard to walk through the woods and not notice it. But you may not have noticed, because you didn't dig it up, the hostorium. So what we have here is a section through the plant, and this is the host root, and then this is the hostorium, this big bulbous portion. But this picture shows they took a hostorium and dissolved away all of the parenchyma tissue. And look at the placental-like arrangement. It turns out that the Canophilus makes the host grow in this way. It's kind of like what in mistletoes we call a wood rose. It co-develops with the host, forming this intricate placental-like feeding structure. So that's pretty cool. So Canophilus has got a couple species. One goes all the way down into Mexico. I actually was on a students committee at Mississauga in Canada. I worked with Sasha Stefanovic, and he did his PhD on molecular systematics and biogeography of Canophilus. So it was a very good study. The other one, Epiphagus, is on beech. And we have that one here in Southern Illinois, and it's all around. I saw it last fall when I was in the Ithaca area. When you find it beech, you usually find the Epiphagus along with it. It has this sort of brown, tan, sort of nondescript, sticky-like stems, no leaves. They're just scale-like if they're present there. And then the flowers are autogamous. Even though they're showing opened up here, most of these flowers have already pollinated themselves. And they make many, many, many little tiny seeds like we see here in this section of a capsule. Epiphagus has the honor of being the very first holoparasitic plant to have its chloroplast genome sequenced way back in the 1990s by Jeff Palmer's lab in Indiana. And it has a tiny little chloroplast genome. Normally they're 150,000 kb, but now this thing is tiny. It's a quarter the size, losing all these genes for photosynthesis. So that got the ball rolling, and now we have chloroplast genome sequences for pretty much all the parasitic plants. All right, time to move into the sandalwood order. This is the group that I spend most of my time studying. You can see its position in the phylogenetic tree here. It's basically sister to all of these asterid groups, like Asteraceae and Apiaceae, even Cornaceae, and so on. So it used to be thought to be a rosid. So this has been bounced around all over with different interpretations, but now we know that it's sort of at the very base down here of the asterids. In this picture, we're looking at the sandalwood of commerce. That's the Santalum album that they distill the oil to use for sandalwood oil, the wood, all sorts of different uses for incense, even medicinals, and a few others. Mistletoe, Phoradendron, and Commandra. These two we're going to talk about because they occur in New York. So again, I've studied the sandalwood order for my whole career, so I have to show sort of the phylogenetic tree here. This is what we know about the relationship of families in the order. The ones at the bottom are non-parasites in white. Green are the hemiparasites, again, yellow are the holoparasites. So there's actually two groups of holoparasites in the sandalwood order. Mistletoes are the ones with an asterisk. Mistletoes are aerial parasites. They occur up in the branches of trees. And you can see that's evolved 5 times independently in the order. And in New York, I've circled the families that you guys have. Commandraceae, you may have known this group right here all as Santalaceae, but my Flora North America treatment has split it based upon the topology of these trees and morphology, as you will see. They're distinct groups for sure, separated by hundreds of millions of years of evolution. All right. So 2 little herbaceous things that occur in New York, one relatively common, the other not so much. Commandra, which has the unflattering name of bastard toadflax, Commandra umbellata. This thing's widespread, occurs all over North America, from Canada to California, all the way to Florida, and even occurs in Asia, or excuse me, Eurasia. There's a little pocket of Commandra over there. And then the other one, Geocolon, much more restricted. It grows mostly in Canada and the tier of states that rim Canada. And you see New York just gets Geocolon. At first, people thought it was another Commandra, but it has a lot of differences. Commandra has a terminal inflorescence versus axillary. The sex of the flowers, Commandra has bisexual flowers. These are unisexual. And the fruits are quite different. Commandra has sort of a almost a nut-like hard achene versus a berry in Geocolon. I haven't yet seen Geocolon. All my colleagues who travel north have seen it. So this is one of my goals, is to get to see Geocolon in the flesh. It was named by Fernald, by the way, the only genus that he named. The other one, and used to be Santalaceae, now we put it in Cervantesiaceae, is Pyrularia, buffalo nut. Okay, you all gonna sing Buffalo Nut, won't you come out to... No, wait, that's the wrong song, huh? Okay. This is one, has anybody seen Buffalo Nut? Okay, you're gonna raise your hand, aren't you? No? Oh, okay, you haven't. Because look at its distribution in New York. Long Island? What? What's it doing down there? It occurs in the Appalachians, Virginia, North Carolina, all the way down through there. So this is the northernmost of its distribution. And I just looked at iNaturalist at some recent photos of it. It sure as the heck grows in Long Island. In fact, the name of the place where it grows, I have it written down here on my other computer. There we go. The West Hills County Park and Nature Preserve. So there's actually nature on Long Island? There must be. I looked at Google Earth and there's actually trees in a nature park, and that's where the pyralaria is. So we need to get it in some other spots, right? It's an interesting plant because of the nut. It's actually quite toxic. These are the nuts here shown immature. You can see here is a mature one. They split open, and apparently you can hear this in the field when they pop open. And they have this very hard endocarp. So this is sort of like a mesocarp, exocarp, stony endocarp type of a fruit. So pyralaria, it has a sister species in Asia, just like another rare North American plant called Buckleya. Buckleya disticophila, Buckleya lanceolata. So these are ancient distributions, right? China, Eastern North America that Asa Gray published on 100 years ago. This is one of them. And it's in a cool family nowadays I call Cerevisiaceae. There's pyralaria, but hey, look at its sisters. Hey, stay there, my mouse, too jumpy. Madagascar, Asia, New Guinea, and Africa. Oh my gosh, look at that clade. And the other clade includes mostly South American things, but I used Commander as my outgroup. All right. All of these things have huge fruits compared to other members of the order. Okubaka is the placeholder. It holds the record. It has a seed this big. It's a huge forest tree in Africa, maybe 4 meters in diameter, 100 some feet tall, and it's a root parasite. And the people know, don't plant your crops around this, because you can see a ring around this tree where it's parasitizing the plants. They don't plant your crops there, you're going to have a poor yield. Okay, so pyralaria is in some very interesting company in that part of the clade, the pyralaria clade, I call this one. So anyway, it's a cool plant to see. Okay, now, finally, mistletoes, the group that I spend most of my time and energy on. Here's the one that's probably most common in Eastern North America, Phoradendron leucarpum, the Eastern mistletoe, occurs all through, Virginia, down through Florida, over to Texas, even all the way out to California. There's been some taxonomic changes. Some people don't agree it's all one species, but the molecular data show that it is. And it did occur in New York. I say did because it's apparently extirpated. Where? Right up there on Staten Island in the very northeast coast. Okay. But apparently people over-collected it for the holiday seasons. And the records show that it was there all over Nissa on gum trees, and it isn't there anymore. Okay, this is from a population near my house here, and I've done some successful inoculations on trees around here to get it growing. So I'm going to talk about that. In fact, why don't I say it now? Let's get it going back at this spot in Clifton. What do you say? We get a few dollars to pay for our gasoline. And why do I say that? Because if you drop down into Joyzy, look at, these are extant populations of Phoradendron that I just checked out on iNaturalist. They're there right now. Let's just bring some seeds right up to here and put them on a gum tree. And by the way, this does work. I have mistletoes all around my yard now. I just put some more seeds on a few days ago. Now's the time. The fruits are ripe in December and January. They're almost past now because the birds have gotten them. December's the best. Well, maybe next year we'll get somebody to do this. Let's get it back in New York. What do you say? Let's get some mistletoe going. And I checked, there's lots of gum trees all over. By the way, those orange dots on the map there are Nissa sylvatica. Okay, so we know we can do this. We can get it going back there. And we'll just use a near population, something genetically similar to the New York ones. And hopefully we won't over harvest it this time. The host range in Phoradendron is broad. This is a partial list of the things that it grows on. But what's really weird about it is it doesn't grow on everything everywhere. It has preferences. So in my neck of the woods, it loves elm and actually red maple. But look at green, New Jersey, it seems to like Nyssa. Virginia, it loves Acer rubrum. And down in the South, it loves oak, Quercus nigra, the water oak. My friends in Texas who worked on this, his student, it's all over the water oaks on the Huntsville campus, Sam Houston State University. So what's going on there? And that's what Chris was working on, is the recognition of the mistletoe and the host. And he actually made progress on figuring it out. It has to do with volatiles that the host gives off. It's a pretty cool study, recently published. So how does it get going? Well, just a quick little bit of what I do. The plant in fruit right here, they're female plants. My ones that I inoculated several years ago made fruits for the first time this year. Yeah. So inside is the fruit wall. You can see here is this viscid or sticky seed that I pulled out. You can see the vicin strands. And when you put it under some backlighting, you can see all the vicin around a green photosynthetic endosperm, and its little tiny radical just beginning to pop out. I stick them in hydrogen peroxide, it makes them germinate. You can then put it on the host, and then it goes bloop, and forms a hostorium and grows right into the host branch, if it's a young enough branch, like 1 or 2 years at the most. And then after a couple of years, you start to get your little baby mistletoe plant growing out like this. These are on trees right on our campus. And again, they got too beautiful, and then people grabbed them during the Christmas season and they're all gone. So I need to get up higher. But we can do this. I want to stick them on Staten Island. Now, the other mistletoe you have in New York is this one, the genus Arsuthobium, called dwarf mistletoe. And this is the one species we have in the eastern US, Arsuthobium pusillum. Most of them are western US and Mexico. But this one was the very first mistletoe described in New York. And a little bit of its life history here, it forms what are called isophasic brooms. These are witch's brooms on the spruce tree. It grows on Picea mariana, the black spruce, sometimes white and red as well. And if you look at this spruce, you can see some mistletoe poking out right in here, but you don't see much mistletoe in here. But actually the mistletoe is already there. Here's the endophyte. It grows all the way up to the tip, and it divides in synchrony with the host cells and is pulled along through development. And then it has different growth phases. So the oldest ones are back here, and then younger and younger. So fruits, flowers, little buds, nothing. Arsuthobium is known for its explosive fruits. They don't need birds to get around like Phoradendron. If you just touch them when they're ripe, the little pedicel breaks off and it shoots the seed out at about 60 miles an hour and lands many, many feet away onto the needle of a host. And then they slide down to the base, form a hostorial connection, and grow in. Then this is the endophytic part slicing through a spruce branch, showing you the sinkers going down into the xylem. So this is all parasite tissue here and here and here. They're separate sexes, male and female. So obviously this is a female plant with the fruits. The little male plants look like that. Spruce needle is about 1 centimeter. So you can see it's a tiny little shoot. Basically, it's just a flower. That's it, little bract leaf. And that's all that pokes up above the surface of the plant. In New York, it shows the distribution here. I need to get out. I almost did it, right, Anna? Marcos and I were going to go out and look for our dwarf mistletoe, but we got busy. So I'm going to go out and see it next year. Have you guys seen it? Has anybody seen the dwarf mistletoe? Yeah, of course. Anna, you've seen it. Well, oops, one more slide here. Come on. There we go. To talk to the Native Plant Society about dwarf mistletoe, I'd be remiss not to mention Lucy Bishop Millington. She was the first botanist to discover it in New York. And there's the map of her area. She actually found it up, I think, right around in here, around Lake George. Okay, she lived and botanized in this area. She was an interesting lady. There's some biographical stuff about her on the internet that one can find published. But she also befriended and corresponded a lot with Charles Horton Peck, who was your state botanist at that time. And he actually named the plant, even though she published her observations in Journal of the Torrey Botanical Club before that. She was the first to describe the explosive seed mechanism. in dwarf mistletoe. So again, at that time, of course, women in botany was a kind of an anomaly. We have to really credit her incredible observational skill. She loved nature. So when you read about her going out to her favorite swampy areas, getting her feet wet and, you know, studying the plants, it's really a case of someone who truly loved outdoors, someone who would probably be a member of the Finger Lakes Native Plant Society, I'm sure, right? So that's all I have. I would say if you want to learn more about parasitic plants, I run a little website called Parasitic Plants, Parasitic Plant Connection, and soon to switch it to Cornell, by the way. I'm going to move it from servers here to a large server suite that Cornell is going to offer us. So, and redo the web page, make it look a little more modern and functional. So lots of information on all parasites there if you have questions. So that's it.
Well, thanks so much, Dan. That's great. We have a few comments in the chat that I can read that might.
Okay. Oh, I see 32 things in there.
Well, let's see.
Millington Brook Bog.
Middleton Brook Bog, yeah.
Was that exactly where she found it?
Well, I don't know. Is that Steve? Is it? That's from Steve Young.
Oh, okay.
And he would probably know.
Not Lake George, but it's near. Just is it the area north of that? I think.
Yeah, it's right near Warrensburg.
Oh, that's right. Whether these populations are still there depends, right? I've looked on Google Earth. Some of them have been developed. They've drained and put, you know, structures up, and I think the landscape in many cases has changed in the last 100 years. And whether they still have dwarf mistletoe, that's an understatement. Yeah. Many people would see it as something negative that you would have to get rid of. I don't know.
Yeah. Steve also mentioned that Schwalbia is extirpated in New York.
Oh.
And was last seen in 1865
Okay.
in the Pine Barrens wetlands. The Albany pine bush, Steve? No.
Oh, I've been to the Albany pine bush. Oh, really?
Yes. Yeah, in Albany pine bush.
Ah, okay.
Yeah.
Lots of questions. Should we start at the top or where do we go here?
Well, up at the top, we've got a lot of things about the quiz, the plant quiz.
Oh, yeah, I see that.
Maple.
Daughter, got it, daughter.
Daughter.
I've been doddering around here.
Says Schwalbia. So Sandplain Gerardia.
It grows in Nassau Communica. Oh, really? Okay.
Yeah.
That's cool.
Let's see. Oh,
Just
so
one loaf.
Liz asked, doesn't mistletoe damage its host?
They do, but after a long time period, right? I mean, really what it does often is decrease the value of wood because of witches' brooms. It diverts the nutrients to those branches that normally self-prune in pines and spruce, right? So they're sitting there with this big lateral branch, like, what's that doing? In Mexico, you see these branches this big around sticking off the side of a pine. It's because it's parasitized. So they really mess with the physiology, keeping those nutrients coming, right? And they don't go through the normal pruning to get a beautiful, symmetrical, unknotted tree. Yeah, they do kill eventually, but not in, I think, natural situations. They live for, 100 years with their parasites. So it's like epiphagus. Does it kill the beech tree? Is it a pathogen?
Right,
I'd say it's a parasite, but it's not really a pathogen, right? Those beeches seem to tolerate it pretty well. So then we get into the idea, is it a commensal or mutual or, I mean, all these other kind of associations. It's truly a parasite. It removes nutrients from the host, but just how damaging it is the question.
Someone else asks, does Commandra parasitize blueberries?
Yeah.
Yeah. Okay.
It's kind of rare in some places. I've been looking for it in Southern Illinois. It's been extirpated from most of the sites here because of development.
Yeah. Here's a question from Alex Petsky. You mentioned 4 genera that specifically parasitize or prefer Fagaceae species. Fagaceae species in other parts of the country or world tend to support relatively many specific parasitic species. If so, do we have an idea why?
Yeah, that's interesting. That Mitrostema, Fagaceae. Now, in the old world, it's, you have other genera, Anglehardias and all these things, but they're sort of Quercus-like things in the Philippine flora. They have acorns, lithocarpus, and all of these, right? And it parasitizes them. So what's the deal? Are they just old and those hosts were available and they got going and sort of canalized onto that? Kevin Nixon, my colleague, is an oak specialist. We should actually talk about that. Why is there this seems to be this preference? And again, the study needs to be done where you quantify the rhizosphere, what's there. I mean, if 90% of the roots are oak, then it kind of deflates the idea that it's, preference is oak, because that's all it has. But what about other cases where oak is just the same as maybe only 5–10%, like a whole bunch of other things? And then if 90% of the parasite grows on oak, then that's something different. That's more significant. But that study we still need to do. A little harder to work in the rhizosphere, right?
Yeah, for sure.
Mm-hmm.
So we got some other questions. Keep coming. Tom Phillips asks, the Agellinus and Oriolaria both have leaves, etc., indicating that they are active photosynthesizers, but Epiphagus seems to be a chlorophyllus. Are the Agellinus hemiparasitic?
Yeah, they're in the hemiparasite group.
Okay, great.
Yeah.
Good. Stephen Daniel wants to know if you're familiar with helosis.
Oh, yeah.
I stumbled across it in Ecuador and wasn't sure at first if it was a fungus or a parasitic plant.
It's Balanopheraceae. It's like back at the beginning here when I showed a Balanophora tuber. This thing, it's in this family with Balanophora. Helosis cayenensis. It's one of the more frequently seen members of Balanopheraceae. Are you guys still seeing my screen?
Yep. Yes, we are.
Okay. Oh, that's cool, because you know what we can do. We'll go there.
We'll go where?
Well, you want to talk about Balanopheraceae? Look, boom, there it is. And helosis.
Yep, absolutely.
There we go. So here's the plant showing emerging from the doodle-doo. Look at that. Whoa, it's a pineapple. No, it's a red. But look at it has hexagonal scales that dehisce. See them laying on the ground? And then the flowers pop out underneath those hexagonal scales.
You're not sharing that screen.
Right. I'm not sure.
Oh, I'm not. Oh,
Right.
sorry.
No, we still see your PowerPoint.
We're seeing the PowerPoint.
So I would have to do something
Just
different.
move the PowerPoint out of the way. Maybe.
No, I think you have to tell it.
You'd need to stop sharing and then start again.
Yeah, stop sharing. And then I share again, and then it asks me where, and then I do this. Now you can see it.
Oh yeah, look at that.
Look at that thing. So those are the hexagonal scales. You can see them up, they're still attached, and then they drop, and underneath are the flowers. It's really beautiful.
It is.
That's a holoparasite, of course. Yeah, it's related to Helosis and Corinia are close relatives in this New World group of Balanopheraceae. Okay. So that's parasitic plant connection. You see lots of Helosis pictures. Langsdorffia, blah, blah, blah.
Wow, yeah, look at that.
Lots of pictures. There's the distribution. So yeah, you see them in Central America all the way down into South America. Corinia occurs there, as well as Helosis. Okay, we had some more questions, right?
Yeah, there's some more. Can you talk a little about the role of horizontal gene transfer for host specialization? Is there any link? Does it make genotyping more difficult? Do we see species being split?
Okay, so that was a topic of when I gave my seminar to the Cornell faculty to become adjunct. I had a whole section on HGT, horizontal gene transfer. I've never seen it connected to the idea of, what did you say, host specificity or something?
Host specialization.
Specialization, or yeah. I've never seen anybody connect it in that way, because a lot of these genes that are moved, we don't really know what's their function. They're often mitochondrial genes. Mitochondria sort of have to do their thing with respiration, and then that's it. It's not like making different kinds of leaves and all of this kind of thing, right? We're beginning to get some information about nuclear genes that have been horizontally transferred. And I think once we have more of that, that's sort of down the road in terms of our understanding of these parasites. Maybe I could answer that, but right now I have no clue about the role of HGT and host specialization. We do know, by the way, that it moves both ways. Parasite genes have been found in hosts. Got that nice plumbing connection, right?
Yeah, of course.
And not just xylem, which is dead, but you have phloem connections, which are living cells. And daughter's been a good study organism to look at the flow of DNA in both directions. So the real question is, how are the hosts keeping all this parasite DNA out of them? Not the other way around, right?
Yeah. Okay, interesting. Couple more questions.
Okay.
Time for a couple more. Where to go? What would you say is the main selective pressure that has led to parasitism? Holoparasites seems low light, but what about hemiparasites?
Yeah, good question. That's been speculated on a million times. So Orobanchaceae is a good example where we have the non-parasite as sister to all those parasites. So the real question I would, if you find some genes associated with hostorial development, could we go ahead and transform the Lindenbergia and turn it into a parasite? What would its roots do if they had some of those genes? Yeah, we're going to make a new parasite of it now. I mean, we don't really know this. I mean, to me, the selective pressure is pretty clear. The fact that parasitism has evolved 12 times independently, by the way, that's extant parasites. Wonder how many experiments went wrong, right? Maybe 10 times that, right? That there's a real advantage to being hooked up to something else and let them buffer the environment. And I'm getting a nice sweet ride, right? Sucking out all the nutrients. So I think there's a trophic push, right? Now, to get yourself into the mode where you have the morphology to do that, maybe micro heterotrophic things are our answer there, because we already know how many plants have associations in their roots with fungi, right? Almost every forest tree does. And those are, there's ones that are internal as well as external. So possibly fungi had a role, and that's a guy named Atsat in California published many papers about this, and people kind of laughed at him, but he may be right. Fungi played a role in that very early establishments of angiosperm connections. It's still speculation. It's an open area.
All right. Let's see. Do you know if any hosts have developed defense mechanisms against the development of parasitic hostorial tissues within host tissues?
Oh yeah, many hosts do. In mistletoes, you see them forming a necrotic layer at the point of attachment. Just basically, apoptosis is the word, fancy word for cell death, programmed cell death. They're being invaded. It happens with fungi too, right? You just kill those cells around it and wall it off. And this happens with many mistletoes. In root parasites, we have the same sort of thing going on. In fact, they're studying it for witchweed because they now have strains of sorghum that do exactly that. The minute Striga tries to attach to its roots, it walls it off. And these basically are resistant to Striga. And you can imagine money to be made with a resistant strain, right, of sorghum to be sold throughout sub-Saharan Africa, where these are subsistent crops, right? People are eating those directly. So this affects people very, very directly. Yeah, so there's a lot of interest. We have a parasite meeting in Nairobi, Kenya this July. I think I'm going to go. It's going to be fun. But, yeah. I mean, in Africa, this is sort of a life and death thing, right? These are not just kind of curiosities. These parasites mean, in Swahili, the word wuta is fire. That's how they describe Striga. It's like your crop has been devastated with fire. They have to move. They can't farm there anymore. It's that bad.
Yeah.
So,
bad.
Billions of dollars lost.
Yeah. One more question, and then maybe, so if people want to contact you, Dan, did we have your
Oh, it's an easy one. Just DN for Dan Nickrent, DN, and then 277 at cornell.edu.
Okay, great.
So just try that email. I have a Gmail address, I have others, but that's an easy one to remember.
277.
That's it, at Cornell.
I'm just going to put it in the chat here so people can see it if they want to
Oh, okay.
contact you. And I think I'll ask a couple more. Well, geez, the questions keep coming. We got time for, we got time for them all.
Oh, we're fine.
Let's see. Oh, so any thoughts? This is an interesting question. Any thoughts or theories why holoparasites like Orobanche still retain their chloroplasts? Is it for heme production or just us witnessing an unfinished process of ongoing evolution?
Yes to both of them. That heme idea, now, that was Ralph Bungard's hypothesis about necessary for the hemes that are mostly in the mitochondrial system, that it was something you couldn't relinquish. Unfortunately, we've done work on Rafflesia-plastome, and it doesn't have one. I don't know how you prove a negative. We haven't published because how do you prove a negative? Molina published their work, but I was co-author on that, but it's still hard to say, have you sequenced every last detail of its genome to know, that you don't have a plastome at all, but pretty good evidence that there's no plastome at all in Rafflesia. Okay, so we have everything from ones that are minor, sort of like Leptria and all these things, Orobanche. I mean, they look like normal chloroplasts, and yet it's a holoparasite, but they're hardly reduced to Epiphagus, that's like a third the size or quarter the size, all the way down to the Balanophoras that we're looking at the screen now. It has the smallest yet. I think it's 12 kb, and it's 86% A and T. It's thrown away GC, it's DNA. 86% AT. What? So what is that doing? I think they figured it's making one or two genes. That's it. Claude DePamphlis used to say it's the tail that wags the dog, right? There's something in there that's still essential, But we know, I think, that it can go the full route, but in any particular lineage, maybe it can't, right? The story may be different in each of those 9 lineages where we see holoparasites, right? Very interesting question, though, right?
Okay. Arceuthobium is one of the earliest flowering plants in New York. In a bog near Rochester, I find it in flower in March. Any thoughts on why? Not a lot of pollinators then.
Ooh. Well, I have a theory. I'd have to be testing this, but it's a neotenal theory. Basically, that this thing, it's a spring flowering. It has to do with when it undergoes meiosis and then actually produces pollen and so on. There's different serums in dwarf mistletoes. There's 4 different ways that it does that. And this is in this other one, spring flowering group, like Arceuthobium vaginatum and Douglasii and so on. But here's the cool thing. When we did the molecular phylogeny of all species of Arceuthobium, published in American Journal of Botany, who's its sister's closest relative? Arceuthobium bicarinatum from Hispaniola in the Caribbean. A great big, tall, dark purpley pine mistletoe is absolute 100% certain the sister to Arceuthobium pusillum. So what's going on? Okay, fossil record shows Arceuthobium pusillum pollen all the way pre-glaciations, all the way down into Georgia. There were spruce bogs in Georgia, yes, and Arceuthobium pusillum, or its ancestor, right? Some version of it was there at that time. Okay, the Caribbean, Florida, maybe we had a connection with dwarf mistletoe ancestor in that Caribbean, southeastern United States flora. I'm speculating here. Who knows what it looked like and what its hosts were? But then came cooler periods, right? And pusillum sort of marched up to the north. Why is it all internal? You think about it, it sticks its tiny little flower out, does its thing, right? And that's it. That's protection, right? Most of the time it's freezing cold. You get your little big bicarinatum shoots frozen off. So that's clearly an adaptation to living in a cold environment. But I think pusillum's an ancient thing that goes all the way back to, I think there's Miocene pollen of Arceuthobium in Georgia. Pretty cool, huh? I mean, who knows what the ancestor looked like and its connection. So did I answer the question? I don't know. I'm just rambling.
I think there's one more. And if anybody else has anything, you feel free to speak up because we got a couple minutes. If the parasite on maize was introduced here from elsewhere, what did it parasitize in its native land, since maize is native to the Western Hemisphere?
Good question. So we're still looking here, right? We can still see. So let's go down to Orobanche. And let's go to hemiparasites, striga, witchweed,
Mhm.
photo gallery. All right. So Asiatica is this one that we were just talking about. But a very close relative is called elegans. This one. In fact, they're so close, some people can hardly tell them apart. Why? When they did the molecular work, they're absolutely 100% sister. They're so genetically close. So Asiatica is an agrostal species. We think that this thing evolved from elegans because of human cultivated crops.
Really?
Yeah, this is a direct result of monocultures, if you will, right? Because elegans never becomes a problem. It's on native grasses in its normal grassland environments, but not Asiatica. Asiatica is the starling, right? That started just spread. You know, it's like starlings may have pretty ancestors and relatives. There are some beautiful starlings, but not the ones that I have in my yard. I keep them away from my bird feed.
Yeah.
So yeah, here's a case where we know that there's a native wild one that is not a problem, and this nasty agrostal called Asiatica.
Right. All right.
Yeah.
Well, thanks, Dan. This has been really, really interesting and great. We had some great photos and some really, some real food for thought. I think, and there's lots of research possibilities here for anyone who's who's on that track.
Yeah, come and see me if somebody does have, an interest. I have thousands of little projects, even citizen scientist type. I mean, like that thing that Keegan Schultz did, just going out and digging up some plants. We now have the method to actually analyze and find out what's going on below the ground. There's a lot of little projects like that that are kind of fun if somebody's interested in doing a little bit of research. Come and see me. I'll be back in April, but you can always email me here. I'm not gone off the planet.
Right. Well, great. Anybody else got anything they want to add or comment? We got a lot of thank yous and wonderful and lots of great info.
Thank you. Thank you much.
Yeah, thank you. Thank you so much, Dan. That's been really,
okay. It was a pleasure.
really great.
See ya.
All right. See you in April. Bye-bye.
Bye-bye.